Hello, everyone, and welcome to today's live broadcast, Proven Answers for Protein, Vaccine, and Gene Therapy Analysis, presented by Dr. Mukesh Malik, Senior Application Scientist, Sciex. I'm Christy Jewell of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Sciex. For more information on our sponsor, please visit sciex.com. Now let's get started. I would like to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer questions at the end of the presentation. If you have trouble seeing or hearing our presentation, click on the Support tab or use that Ask a Question box to let us know you're experiencing a problem. This presentation is educational and does offer continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I'd now like to introduce our speaker, Dr. Mukesh Malik. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Malik, you may now begin your presentation.
Thank you, Christy, for that introduction. My name is Mukesh Malik, and I'm the CE Application Scientist at Sciex. And I'll be talking about analysis of protein, vaccine, and specifically gene therapy analysis using Sciex system. So I'll be covering capillary electrophoresis, introduction to capillary electrophoresis, and what are the practical applications of CE. Then we'll be going into oligo analysis, where specifically we'll be talking about plasmid analysis, double-stranded DNA, single-stranded DNA, and RNA analysis. Then we'll jump into viral vector analysis, and then we'll just summarize these analyses. So what is capillary electrophoresis? Capillary electrophoresis is the movement of charged or polar molecules inside a capillary filled with conductive fluid under the influence of an uniform electric field. So we say at Sciex, if an analyte is charged or polar, look to CE first, because you can easily analyze any of these molecules by CE. Okay, so let's talk about analytical benefits of CE compared to LC. In CE, there is differential migration of molecules in an applied electric field. It's electrophoresis, which is orthogonal to LC. It's not replacement of LC. In CE, you have exceptional resolving power and peak efficiencies. You can get more than 1 million theoretical plates. If you look here, a cartoon here on your right bottom, if you have about 4,000 plates, the peak width is really wide. But if you increase the theoretical plate, which we get in CE, to about, say, 90,000 or so, which is the filled peak here, you get a nice sharp peak here. The other advantage of CE is small injection volumes and minimal run buffer requirements. And with CE, you get separation flexibility. You can do capillary zone electrophoresis, capillary cell electrophoresis, and you can have selection of different kind of buffers for separation capabilities. And the other thing which is very nice is its automated quantitative technique. You can use CE for characterization for a wide range of molecules. You can analyze single-stranded DNA, double-stranded DNA, plasmid, antibodies, or even viral particles. For gene therapy, to make the virus, so you start from 2 to 4 plasmids and you transfect the cell line with that, and then in cell culture, depends on the plasmid set you use, you get either adeno-associated virus, lentivirus, or adenovirus, and you package within these viruses genetic material based on the plasmid you use. And you can use this now the virus particle where you package the DNA either for ex vivo gene therapy or in vivo gene therapy. In ex vivo gene therapy, then you transfect the cell line outside the patient, and then you take those cell lines back into patient. For in vivo gene therapy, you transduce this virus particle, which is containing the gene of interest, into the patient, and the transgene get into the patient and expression there. Now, like I said, so viral vectors are common delivery vehicles, and these viral vectors are composed of proteins and nucleic acid. The protein is the outside capsid coat proteins of these viral particles, and nucleic acid is the genetic material which is packaged within the capsid there. And CE is the ideal tool for analysis of both the nucleic acid and proteins. Now, because the starting material is the nucleic acid, so first thing we have to do is we need to do quality assessment of nucleic acid. But for nucleic acid quality assessment, the genetic material used in gene therapies are plasmids, like I said before, or viral vector products. For plasmids, the genetic material which is delivered into the cell to replace or inactivated disease-causing gene or introduce a new gene to help treat a disease. You might have mRNA to supplement or replace expression of protein to correct a function associated with a disease. You might get small oligonucleotides, which are small nucleic acid that regulates gene expression and protein expression, like siRNA, which is small interfering RNA kind of things. You need to do quality assessment of these genetic materials for product safety and efficacy. And what kind of analysis is you can do? You can analyze plasmid isoforms, messenger RNA purity and degradation. You can do oligonucleotide size and impurity determination, and you can analyze vector loads. So let's get into oligo analysis, which is plasmid DNA and RNA analysis in a little bit more detail. For plasmid quality and structure analysis, you have the plasmid sample here, and then you use Sciex P800 Plus system, and we have a kit to analyze this, which is the dsDNA 1000 kit, and you can, for detection using LIF, you can use an intercalating dye, which is enhanced dye, which is an intercalating dye. And this cartoon is just showing what kind of separation you get. On right side, you can see a baseline resolution of supercoiled to open circle here. And let's get into detail of that little bit. So if you look here on the left side, so at the bottom here, A is what a typical plasmid prep look like. This is a 7 kb plasmid here. And how do you then identify which peak is which peak, if these peaks? So if you do restriction digest of your plasmid, you get the linear peak, which is in B here in this cartoon. And if you use a enzyme which just cleave at single strand, so you get a nicked, which is the open circle, you get this peak, and these minor peaks are like some impurities within the enzyme which linearize the plasmid a little bit, and this that's what those extra peaks are. But this is the nicked plasmid peak here, and this is a mixture. So that's how you can come to know, like, this is your supercoiled peak right here, and this is your open circle peak. And this side, on the right side, is a separation 18 runs of a 5 kb plasmid typical separation, but just to show you the reproducibility. And here at the bottom table, as you can see, you can get less than 1% percent RSDs for migration time and percent area. So it's a very, very reproducible area. And why you need to do plasmid separation? FDA guidelines recommend that your plasmid has to be more than 80% supercoiled. That's why this requirement for plasmid analysis for determining the percentage of supercoiled to other impurities present within your plasmid prep. You can use the same double-stranded DNA kit for analyzing if you want to do restriction fragment analysis or other fragment analysis of your plasmid or other DNA. And this is the cartoon just showing that you can do from about 50 base pair to more than 1,000 base pair separation. And here this cartoon is just showing you that you can get separation from 271 to 281 base pair baseline resolution here. And the second thing is, like, if you are using UV detection, this is about 200 microgram per ml or 200 nanogram per microliter. But if you use LIF detection, you can easily detect at 10 nanogram per microliter or 10 microgram per ml, and very nice signal there. We can use the same kit, slightly different separation conditions and different capillary length, and we can extend the range. We can separate from 50 nucleotide, or in this case, the data is from 72 nucleotide, 72 base pair, all the way to 15 kilo base pair. So you have a very big range of separation where you can do fragment analysis, or if you are doing restriction fragment analysis of your plasmid. And here you can see these different color codings are the fragments of DNA came from different sources, like here. So they came from DNA Ladder or PUC18 plasmid or 5174 bacteriophage. So the source is different for each different peak here. But as you can easily see that we can do separations, baseline resolution between 271 and 281 here, and then 3 kb is here. So there is a baseline resolution you can get here. If you have single-stranded DNA, small oligonucleotide, we have a kit for that also, which is ssDNA 100R kit. And here you can use this kit to analyze or oligonucleotide, which are single-stranded DNA fragments from 10 to 100 bases. By this kit, you get single base resolution. And we use replaceable gel buffer here with coated capillary for maximum reproducibility. If you would like to study the oligonucleotide having secondary structure and you would like to study that, you can reconstitute the gel without urea to preserve the secondary structures here. This is a electrophoregram showing up separation of poly A 40 to 60 bases, which is standard included with the kit. And as you can see here, there is baseline resolution for all these 21 fragments here. And the percent RSD for both migration time and peak area is less than 0.5% using this ssDNA kit. You can use the same kit, ssDNA kit, for small RNA or DNA, like I said, single-stranded DNA up to 100 bases. This cartoon on the left here, electrophoregram, is for a siRNA, which is 10 bases long. As you can see, the major peak and some minor impurities present within the sample to determine the purity of your siRNA prep. This on the right side is a 45 base long ssDNA fragment, and as you can see, there are some N-1 and 2, N-2 and -3 kind of fragments, so 45, 44, 43, and so on. So what kind of impurity you have in there. You can use the same ssDNA kit for analysis of modified oligonucleotide also. This is one showing TRITYLATED phosphorothioate oligonucleotide. And here on this here, this is a crude sample on your left side, which is B picture here, and you can see lots of impurities present in addition to the oligo of interest. And this is a failed sequence where we didn't make the oligonucleotide correct, and you see lots of impurities there. And this is the final product pool, and you have the major peak here, and there is a minor impurity present in the sample there. Now, for RNA purity analysis, we have method for that also using P800+ system, and this method is used for RNA purity determination, and we get baseline resolution from 200 bases to 6.5 kilobases. We use the P800+ and LIF detection here, which is default 488 excitation, 520 emission, and we use the pre-assembled easy cartridge here. Now, if you look at the data, this is the resolution here. So these from 200 bases to 6.6 kb here is the marker, which is thermo RNA 6000 ladder. And 1.2 is the spike in peak here. And as you can see, there is a very nice baseline resolution from 1 kb to 1.2, and this is a 2 kb peak. And this one on the right side, the electrophoresis showing the repeatability from 8 different injections. So you get very, very nice reproducible result here. Now, this is like what kind of sensitivity can we achieve? So we can get LOQ of 0.33 nanogram per ml and LOD of 0.08 nanogram per ml. And this is determined based on the spiked 1.2 kb fragment, which is spiked within the marker. And you can see the relationship is very linear for a long dynamic range. So just to summarize, we have tools for both RNA and DNA analysis, and using our P800+ system. For RNA and DNA, it's automated method versus manual slab gels, and you get much higher resolution than what you get by HPLC. For plasmid, the CE method correlates with analytical ultra method, and HPLC may not correlate with AUC. CE method correlate with agro gel method also, which is the manual slab gel. But using CE, you get the automated method here. Analysis is in minutes versus days if you are using AUC. It's simple and economic, and the system is much more flexible for multiple molecule types and methods. It's the same system, so it's kind of an open platform. Now, once you have analyzed your genetic material, plasmid, and you package it into viral vectors, then you need to analyze the viral vector. So first thing you have to do is viral vector capsid protein analysis. So AAVs are the most commonly and widely used genetic delivery vehicle for gene therapy presently. And the reason for wide usage of AAV is they are non-pathogenicity, low immunogenicity, and differential tropism in multiple cell types. Basically, you have different serotypes which can target different cell types. And without going through detail, so you need to analyze purity analysis of AAV viral capsid proteins for quality assurance and safety of AAV products, that you made the right viral vector. We used for capsid protein purity analysis and size analysis, we use the standard SDS molecular weight analysis kit, which is kind of one of the workhorses which is used for on P800 plus system. It's identical to SDS page. Uses generic methods. It's direct quantitation if you use PDA detector, which is the UV detection. You can use laser-induced fluorescence for increased sensitivity after labeling your proteins with a fluorescent dye. The methods are automated here. So for capsid purity using UV, you do the sample denaturation after adding sample buffer which contains SDS and heating the sample. Then you run the sample on P800 plus and using the SDS molecular weight kit, and the cartridge is pre-assembled easy CE cartridge, and the detection is using PDA. So it's UV detection at 214 or 220 nanometer you can do. And this is just to show a cartoon electrophoregram showing what kind of separation you get. And you have three major protein, BP1, 2, 3 here. And if there are impurities, you see them also here. And like I said previously, if you would like to increase sensitivity for this capsid protein determination and presence of impurities, you can do label the proteins with the fluorescent dye after denaturation of the sample. And there are other dyes also available. This data is based on FQ dyes, and again, same thing, and you do LIF detection here. You have P800 plus, but the kit is the same SDS molecular weight kit, and the cartridge is Easy C cartridge. And you get this VP123, similar to what you get with UV, but increased sensitivity here, much higher sensitivity for impurity determination. This is capsid purity, just to show you example for different serotypes. So we have AV8 on the left and AV2 separation on the right, and this is 8 injections in each one, and you can see reproducibility of the data, and you can determine impurities and if there are other proteins present there within the caps, viral prep you have. And this is to show like what kind of viral particles you need. So this is linearity showing that you can, and this is just by UV, and we did determination for VP3. So you can get from 5 into 10 to the power genome count per ml to 10 to the power 14 genome count per ml. And as you can see, it's very, very linear graph here. So you have a wider range just by UV. And if you use the LIF, your lower end will further expand there. Now, if you are looking at empty full capsid, this is a cartoon showing empty full separation using CE-LIF, this one. And as you can see, this side, this peak is empty, some partially filled. This is full peak here. And the top one is empty and rich AV prep. And the bottom one is the enriched for full here. And Sciex is currently looking and developing novel CE-based separation for AV capsid empty full analysis for differential separation of that. And we don't have methods, but we are partnering with customers to develop custom methods for different serotypes of AV and sample formulations. And we are looking at method from both capillary zone electrophoresis and CE-LIF for separation of different serotypes. But we are looking for partnership with customers at this time for empty full analysis. Now, I'll just briefly touch on, we have like for genomics also for gene expression. So we have a system called Genome-led GEXP, which you can use for the genomics for sequencing and qPCR. So this is one system where you can do the DNA sequencing, and the DNA sequencing is using Sanger sequencing here. You can do quantitative gene expression study also, and you can do genotyping and SNP analysis. You can do STR analysis and DNA profiling using this Genome-led GEXP system. Just to conclude what we have learned so far, what all we can do. So we have diverse CE solutions for cell and gene therapy. And what all applications I showed you we can do at this time is plasmid topology and purity analysis. We can do double-stranded DNA analysis, which is fragment sizing. We can do oligonucleotide, which is like single-stranded DNA. We can analyze RNA for purity determination. We can do purity of AAV viral capsid proteins. And you can, using GXP, you can do quantitative gene expression, DNA sequencing using Sanger sequencing, and genotyping and SNP analysis. You can do STR and DNA profiling. Now, this is what system we use. So this is our P800 Plus, which is the workforce for all these analysis I just showed you. And we have different kits, like I was mentioning during the talk, for different analysis or different methods. So this is the system P800 Plus, which is robust, reproducible, and automated analysis of pharmaceutical we can do. There is sample temperature control in this system for sample stability. And we can control the capillary temperature for robust migration time reproducibility. This system P800 Plus comes with interchangeable 3 different modules for detection. You can get, you can have either PDA detection, which is photodiode array or diode array detection. You can have laser-induced fluorescence detection, or you can have UV detection. And we have the easy, for some of the application, easy CE, which is a pre-assembled cartridge having bare fused silica capillary for some of the typical applications. And I would like to acknowledge some of my colleagues who have helped with the data shown during this presentation: Ting Lee, Jane Luo, Elliot Jones, Andy Eonto, Sahana Mola. And thanks for your time for listening to this webinar. And now I'll take any questions you have.
Thank you, Dr. Malik, for your informative presentation. We will now start the live Q&A portion of our webinar. If you have a question you would like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we will answer as many of your questions as we have time for. Okay, Dr. Malik, let's start with this first question. Can we use other dye besides FQ for capsid protein labeling?
Hi. Yeah, we are working on some other dyes where you don't have to use potassium cyanide. Like FQ, you have to use potassium cyanide during labeling reactions. And we don't have to do cleanup also. So we are working on some dye, and we should be coming out with a technical note for these viral capsid protein labeling with that new dye very soon. Maybe like before end of the month, hopefully.
Thank you. Our next question, up to what size plasmid can you analyze?
So the plasmid analysis we have done using double-stranded DNA kit. We just dilute that gel to do the plasmid analysis. So up to 7 kb plasmid, the electrophoregram data I showed you. we did 1:10 dilution of that gel. And I believe we can run more than 7 kb even with that dilution, little bit longer, but for little longer plasmid, we can change the capillary length. We can have little longer capillary length and maybe a slightly different dilution of the gel, and it's 1:10, and we can achieve much longer plasmid separation for their topology analysis, purity analysis of plasmid.
Very good. Thank you. Our next question, can you distinguish between empty and full viral capsids on this system?
That's $1 million question. Like I said in my slide, we have, depends on like the samples we had, like some commercial samples, some kind of like what we feel we are getting it, but we haven't analyzed all different serotypes, so it depends on serotype and samples and what kind of thing. So we are not there to release like that. This is the method, like we have methods for plasmid or CE-SDS, viral capsid protein. So empty full analysis, we are not there yet. But if someone is interested in that one, we are willing to collaborate and trying to work with you, trying to see how we can analyze your viral empty full analysis using both CE-LIF and CE methods. So if you are interested, you can either contact your local application guy or maybe your sales guy, and we can just take it from there, and we can start the kind of a collaborative method development process from that. And we just don't have a kind of like go-to method at this time that, oh, here is the method and just run for every serotype. But we have some working methods little bit, so we are open for collaboration for that kind of analysis at this point of time. Does that answer your question?
Thank you, Dr. Malik. Now, do you have any final comments for our audience?
Yeah, thanks everyone. And if you have further question or even for empty full analysis, if you guys want like something, like I said, you can contact either your application guy or sales guy, and we can take it from there. And if you have any other questions, again, you can come through either of those, either local application person or the sales folks, and we can answer your questions, or you can go to our tech support and they can forward those questions to us too. And thanks everyone for your time.
And thank you, Dr. Malik, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Sciex, for underwriting today's educational webcast. We'd also like to thank our audience for joining us and for their interesting questions. Questions we did not have time to get to today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. Now, this webcast can be viewed on demand, and you can submit questions. LabRoots will alert you via email when it is available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. You will now be redirected to the registration page of our next Sciex webinar, a sensitive and robust plasmid analysis method by capillary electrophoresis with laser-induced fluorescence detection. Presented by Colleen Reed, broadcasting live tomorrow, December 11th at 7 a.m. Pacific Time, 10 a.m. Eastern Time. And we hope to see you there. Have a great day.